Hello, my name is Kaitlyn See, and I am an undergraduate researcher at Montana State University, and today I would like to introduce to you the project that I am currently working on. I work in the cell biology and neuroscience department, focusing primarily on cell signaling in early embryonic development. I am studying the role of an aquaglyceroporin, aquaporin 3b, during early gastrulation and how it aids in the coordination of convergent extension, a process in which cells fold into the interior of an embryo and intercalate to form a long, narrow band of cells within the interior. Aquaglyceroporins are an integral membrane protein channel that allow for the passage of water, glycerol, and a few other small uncharged molecules across the cell membrane. These have been well studied in regards to their function in maintaining homeostasis of a cell, though little is known about how they contribute to intercellular signaling. I have observed that when aquaporin 3b is inhibited, defects are present in convergent extension. In order to study this process, I use Xenopus laevis embryos as a model system. I can inject a morpholino oligonucleotide which binds with a complementary sequence on aquaporin 3b mRNA to prevent the ribosome from binding and translating the gene. This effectively prevents the expression of aquaporin 3b. I can also inject the mRNA for a protein that is involved in Wnt signaling pathways to bypass previous steps that are not activated due to the absence of aquaporin 3b. If normal development is observed, then it can be determined that aquaporin 3b acts through that pathway. As convergent extension is primarily an internal process, I remove the dorsal portion of the embryo at the beginning of gastrulation, indicated by a crescent-shaped swath of pigment which accumulates at the dorsal lip of the blastopore. The removed section of embryo is then cultured under a cover slip to cause the explant to produce an outward projection rather than curling into itself. This allows me to easily determine if defects are present in convergent extension. So far, I have been able to tentatively say that aquaporin 3b acts through a non-canonical Wnt signaling pathway, as I was able to observe normal function when mRNA for disheveled 2 delta Dix is introduced into the embryo, while aquaporin 3b is inhibited. I am currently working on rescue experiments involving PKC, CDC42, and RAC1. It is too early to make any concrete claims, as further experimentation and proper statistical analysis is required. I believe that this research is worthwhile, as aquaporins and aquaglyceroporins are ubiquitous during development, as well as within adult tissues. Understanding how these channels function within a tissue can possibly lead to the development of better treatments for diseases and disorders that are caused by defects in aquaporins and aquaglyceroporins. For example, hereditary nephrogenic diabetes insipidus, which is caused by a defect in the expression of aquaporin 2. Thank you for your time and consideration. I hope you have a great day.